All right, everyone, it's just about 4 o'clock, so we're going to go ahead and get started. So good afternoon and welcome to the fifth concurrent session of the day, Neurotransmitters and Communication Between Neurons. Our presenter today is Dr. Laurie Slifka from Keiser University. Dr. Laurie Slifka is research neuropsychologist with specific expertise in health conditions such as human immune deficiency virus and early life stress that primarily impact brain structures deep beneath the surface of the cerebral cortex. A majority of her research studies incorporate neuropsychological tests and structural neuroimaging. She has also developed research protocols and statistical analysis plans, designed neuropsychological batteries, and created databases for integration of clinical, neuroimaging, and neuropsychological data. She has been funded by the National Institute of Health for her work investigating brain integrity in individuals with human immune deficiency virus. Additionally, Dr. Slifka has contributed to numerous peer-reviewed publications and presented work at international conferences. She currently serves as an advocate reviewer for several peer-reviewed journals, including Psychiatry Research, Neuroimaging, Behavioral Brain Research, Journal of the International Neuropsychological Society, and Neurobiology of Aging. Dr. Slifka has a passion for teaching and utilizes her abundant training and research experiences to inform and greatly enrich her teaching and mentoring practices. If you have any questions for the presenter during the session, please feel free to enter those into the Q&A section at the bottom of the Zoom screen, and we'll address them at the end of the talk. On that note, I'm going to hand it over to our presenter.
All right, thank you so much for the warm introduction, Brian, and sorry about the scrolling through the slides. You'd think after a year of being here in my home on Zoom, I would have a better grasp of Zoom, but it's
You're all
Friday.
good. Take it away.
Huh. Can you hear me okay?
Yeah, we can hear you.
Okay, yeah, so it's Friday, so hopefully everybody gives me a little bit of break on my lack of technological skills today. So Brian, thanks again for the introduction. And thank you everybody for coming and spending your Friday afternoon with me to talk about the brain, which is my favorite thing to talk about. As Brian mentioned, we're going to be talking about neurotransmitters and communication between neurons. You may think, why is this girl talking to me about these things at an education conference? Well, neuroscience is incorporated in the intro psych curriculum, and many of you may teach intro to biopsych. And this can be a really tough topic. When I was first learning this as a student, and then when I was first teaching this, it took me a little while to grasp this, especially before I went into the field. And I know students can often be very intimidated when you start throwing neuroscience at them. So hopefully this can serve as a model for you, and I can give you some ideas of how you can incorporate this information in your classrooms. And then hopefully I can teach you a thing or two. For many of you, this may be a review, but I do hope you learned something. So Brian did a really nice job providing the introduction. Just to brief over a little bit about me, I have my PhD in psychology with an emphasis of behavioral neuroscience from the University of Missouri, St. Louis. I currently live in Chicago, Illinois with my family. My husband and I recently welcomed a baby girl, so it's been a lot of fun these past 7 months being able to work from home and spending time with her. I'm a professor of psychology at Keiser University. The location of the university is actually in Florida. I am fully remote now, so I'm working out of Chicago. I teach both master's and PhD students that are enrolled in the psychology, general psychology program and industrial organizational psychology program. I hate saying expert because I think it's very difficult to ever become an expert in something, but I do know a lot about the brain in individuals with HIV who've experienced early life stress and aging individuals. What all of these have in common is if you flip the brain over, that area is called the subcortex. All of these different conditions, aging, it's a bit odd to call aging a condition, but they essentially all affect this area of the brain. So I study this by using neuropsychological testing, which is cognitive tests. So tests of thinking, memory, executive function, motor skills, and also neuroimaging. There's both structural and functional neuroimaging. I focus on the structural neuroimaging, and specifically a technique called diffusion tensor imaging, which maps the pathways of the neuronal connections throughout the brain and looks at the integrity or the health of those connections. And that's what we're going to be talking about today. So that's taking it a step further from what we're doing today, but it kind of all comes together very nicely. I know you all probably do this too. I am guilty of using the same jokes over and over again with my students. So those that have had me many times were like, oh, she's doing it again. But I wanted to provide you with my research profile in case you have any interest in neuroscience. I always tell my students if they have insomnia, they're welcome to read my research and it could help put them to sleep. So I'm reusing that joke with you as well. Alrighty. So first, I like to provide an overview so you know what's to come. I have provided some talks for Hawks in the past. I've specifically focused on communication within neurons, and I've also done one of communication between neurons. Communication within neurons serves as essentially a building block for understanding communication between neurons. If you did want a refresher on that, feel free to check out the Hawks blog, and there is an archived video on that. So for those of you that have attended that talk before, then you've seen that information. But we're going to quickly go over that again. So we're going to go over some information on communication within neurons, and then we're going to jump to the main focus of today, which is the connections between individual neurons. And then we're going to talk about neurotransmitters and receptor types. We're going to go kind of into like a basic overview of neurotransmitters and receptors, something that you may do in an intro to psych class or an intro to biopsych class. So we're not going to go so deep into the weeds of that, but hopefully it gives everyone a general understanding. All right, so like I said, we're going to start with a review for some of you, maybe new information for others of you, of communication that takes place within neurons. So if I were in a traditional classroom, I would say, what is the term that you associate with communication within neurons? It's two words. I'm looking for the specific term. I'll give you a second to think about it. You can type it in the chat if you want. All right, so I'm thinking of action potentials. Oh, I just see that. Q&A. All right, so I do see a question, and I'm happy to address that one at the end when we go through all of this. So Stephen, I will get to that question at the end. So what I'm thinking of, synapse is involved in it, and synapse is really involved when we think of between neuronal communication. But the word that I'm thinking of, or the term that I'm thinking of right now, is action potential. Okay, so that's really what's taking place when there's communication taking place within an individual neuron. And I see that there are questions. I'm happy to address them at the end. So what you see here is the typical picture of a neuron. So if you were to Google neuron, this would probably be what comes up. It's a very simplistic visualization of a neuron. There are all different types of neurons, but for the purpose of this talk, we're going to stick to this model. So I like to, there's three main parts of the neuron. There's others, but there are the three that we really like to focus on when we're talking about communication: the dendrites, the cell body, and the axon. So I like to use with my students a hand model. I always look really silly doing this on Zoom, but essentially if I have my hand sticking out and I have my arm, the dendrites are my fingertips, okay? And the cell body, soma, would be my palm, and then the axon would be my arm. So I may go back and refer to this throughout the talk. So let's briefly go over the functions of each of these regions. So let's start with the main energy center. What I need right now, some energy, right? Bad joke again. So the cell body, aka the soma, it's the factory of the neuron. It contains the nucleus, which is extremely important, contains genetic information, including information for cell development and synthesis of proteins necessary for cell maintenance and survival. Within that nucleus, one really important area is mitochondria, which produces energy to fuel cellular activities. This area, the cell body, is surrounded by what's called a cell membrane, also known as a neuronal membrane. This is going to be important because this is going to help explain what happens when an action potential takes place. But essentially, this area serves as a barrier to enclose the fluid within the cell body. And so this keeps the good things in and the bad things out. So it keeps certain ions and small molecules out of the cells while letting some in. Accumulates nutrients and rejects harmful substances to keep the brain safe. Establishes an electrical potential within the cell, which will be relevant in just a bit, and is important for conducting an impulse, which will also be relevant in just a bit. Our fingertips that stem from the cell body or soma are the dendrites. I like to think of that as the listening end. I learned that actually in graduate school from a professor, and I've carried it with me. These structures, the listening, or the dendrites, the listening end, they're listening and they're receiving information from a neighboring neuron. So the dendrites are the main apparatus to receive signals. And then the axon, which is my arm, so it's that long structure that's in the model that you're seeing. I like to think of that as a talking end. So that primarily communicates with other dendrites. So at its most basic level, it's a tube-like structure that carries the electrical impulse from the cell body to the structures at the opposite end of the neuron, specifically axon terminals. That's where we start passing information on to neighboring neurons, and that's what gets into our communication between neurons. The axon is covered, most axons are covered by what's called a myelin sheath, which is a fatty material to help speed up signals. So it's like kind of think of it like as an oil, essentially, that's covering it, that makes it faster as a signal is taking place. The nodes of Ranvier are in the diagram that you're seeing. They're a periodic gap within the myelin that helps speed up the impulses. The impulses are able to essentially jump from node to node that speeds up that communication. And then we see the axon terminal, which is at the end of the axon, and that's where the signals are being sent from the presynaptic neuron to the postsynaptic neuron. All righty. I do see those questions are being asked, and I do appreciate that you are asking them, and please continue to ask them, and I will certainly address them. Okay. So let's talk a little bit about the route of communication within neurons. So again, we're still within that individual neuron. So again, this is what a typical neuron may look like. There are general classes of neurons, but all neurons have three basic functions. They are to receive information, typically through dendrites. They are to integrate incoming signals and determine whether information should be passed along or not. And they also communicate with target cells, so other neurons, and then in other cases, muscles or glands, which for the purposes of this talk, we're not going to get too deep into that, but know that there is communication that takes place to other structures aside from just neurons themselves. So just to start us off here, in terms of communication, this is a very elementary explanation, but we're going to go into more detail. An action potential is what the communication is, essentially, that takes place within an individual neuron. It occurs when information is sent down the length of an axon away from a cell body. Neuroscientists may use the word spike or impulse for an action potential. And essentially, it's an explosion of activity that's created by what's called a depolarizing current. So the cell becomes more positive. Hold tight. So what do I mean when I say the cell becomes more positive? So let's go back to when we talked about that cell membrane, that coating over the cell body that helps keep good things in and bad things out. It also allows for certain ions to get into the cell and out of the cell when needed. But at rest, what is maintained within the cell is a negative concentration of ions. So when the cell is not firing, no action is taking place, the inside of the neuron is in negative compared to the outside. So at rest, the cell is negative. So what we typically see is potassium and organic anions are typically found at higher concentrations within the cell than the outside. And then positively charged sodium and negatively charged chloride are typically found at higher concentrations outside of the cell. So this value is not too relevant, but it brings home the point that the cell is negative. So there's variation within an individual cell, but typically we see a cell at rest in between -50 and -75 millivolts. When an action potential takes place, that cell becomes more positive. This balance that keeps everything in check, this homeostatic mechanism, is helped maintain by something called the sodium-potassium pump. And that pushes sodium ions out of the cell and potassium ions into the cell after an action potential fires to maintain the balance of chemicals. And so that really helps after the action potential fires. That's a really quick process, then everything has to go back together to get ready for a potential other action potential. So this is pretty much in every intro psych textbook. And I know when I was first learning about this, I was like, this is complicated. I didn't want to go in this heavy science field, is what I thought at the time. Like, why do I have to learn this? I really don't understand it. But when you really break it down, it, at least for me, tended to make more sense when you really look at the individual parts. And I think that's really important for the students to really break it down and to provide them multiple resources for understanding this. So verbally explaining it, giving them, obviously information, the textbooks, sometimes supplemental videos and whatnot are helpful too. So essentially, like I said before, an action potential occurs when a neuron sends information down the axon away from the cell body. Okay, this is what we see at the micro level. I had mentioned before that sometimes we refer to an action potential as an explosion of information. And this creates what's called a depolarizing current. This means that some event or stimulus causes the resting potential of the cell to move more positive. So to go towards 0 millivolts, when we know at rest, it's between 70 and -55. So when the threshold level is reached, so if it gets to the threshold, say -55, that threshold is the threshold for that individual cell, then an action potential will fire. If all action potentials are a fixed size. So if the threshold of excitation is reached, an action potential will fire, okay? So let's break down these individual steps. So in short, action potentials are caused when different ions cross the cell membrane, or that barrier outside of the cell. A stimulus that we're going to go into when we talk about communication between neurons, but essentially a stimulus that's typically sent from another neuron, if it's strong enough and allows the cell to reach the threshold of excitation based on the movement of the ions, what happens is this causes sodium channels in the cell to open. Because there are so many more sodium channels on the outside, the inside of the neuron is negative relative to the outside. The sodium channel or sodium ions are going to rush in the cell. Okay, so remember, sodium has a positive charge, so the neuron becomes more positive and depolarizes. It takes longer for the potassium channels to open, so that comes after that. When they do open, potassium rushes out of the cell, reversing that depolarization. Also, at about the same time, and this is all happening like this, lightning fast, the sodium channels start to close, this causes the action potential to repolarize. So essentially to go back to -70 millivolts. The action potential actually goes back it goes further negative than -70 millivolts before it evens out to like that homeostatic level again. This process is called hyperpolarization. So it becomes even more negative because the potassium channels stay open a bit too long. But gradually, based on the mechanism of the sodium potassium pump, the ion concentrations go back to that resting level of -70 millivolts. This starts the action of the communication that takes place between the neurons. Okay, so we get to a point after this that information sent to the axon terminals, which we saw a picture of, and then we start this process essentially in another neuron because of the communication from, say, this individual neuron to another. So you may have seen an image that looks like this. It's a very popular image to see. People love showing off neuroscience images, especially ones that look really cool. I did see one comment already about fMRI and like reading minds. I'll address that, and I don't have that exact question up, but I think people take this stuff and run with it. I don't think fMRIs can help us to read minds. I don't think people that have a clinical psychology degree can read minds either. But people love brain images. So you've probably seen this all over the place and be like, what is that? Or, but it's an image of a diffusion tensor imaging scan. And honestly, this doesn't tell us that much. It gives us directionality of the individual pathways within the brain. That's different colors. It can be used, I'm going on a tangent, but just for fun. This can be used as like surgical maps, but if we're studying the brain, this doesn't give us much about what's going on at the microscopic level. It just provides a really nice picture for us. It's also the cover of a Muse album from years ago. So fun fact. But this picture is telling us about the directionality of the communication, the flow of all of the action potentials taking place together, which way they're going. And essentially, these are pathways of neurons communicating with one another. I like to describe this communication system that takes place like a highway system. So in our brain, we have gray matter structures that are essentially our hubs, like our amygdala, our hippocampus, hypothalamus. And then we have communication or pathways that facilitate information from one structure to another. So I used to live in South Florida, and I honestly know the geography better because I've been in lockdown essentially since I've lived in Chicago. But if you're familiar with South Florida, the main cities in South Florida are Palm Beach, Fort Lauderdale, Miami. So those would be like our macro structures, like the hippocampus, amygdala, and hypothalamus. And then 95, which is the most annoying, horrible highway in the world in South Florida, takes us from each one of those cities. So this is essentially what we're looking at here, and essentially what's going on when we talk about communication that's taking place between neurons. I think just for the focus of neuroscience, what's really cool to think about when we actually break this down, like the science of it, we break down the communication that's taking place between neurons, like what's really happening. What's really happening is everything. So all of that communication taking place is what is guiding us to do everything that we're doing in every moment. So right now I'm talking, I'm breathing, I'm remembering information from the past. I'm using my hands too much because I always do. My boss jokes, but he's like, you're conducting an orchestra. I'm like, yep. And then what you're doing on your side of it, you're listening, you may be writing something down, you may be listening to your spouse talk in the back. We all have a lot going on right now, but all of this is happening because of the coordination of the signals that are being sent all throughout our brain constantly. Okay. So with that segue, let's talk a little bit about the communication that takes place throughout the brain. Alrighty, I'm getting distracted by the questions, but I am going to say them at the end, till the end. So communication between neurons, also called synaptic transmission. All right. So what we're talking about for simplistic sake here today is communication that takes place between two neurons. So if you can think of two neurons in your head. I've shown you some images of them when I describe this. This is what you should go to. Two of the hand models would be like that together. Kind of silly, but they're essentially like the dendrites receiving information from a cell down the axon and then sending information to different neurons to the dendrites, which are the listening. So we're going to, there are a lot of different classes of neurons, but we're going to refer to this typical neuron, the image that I showed you before. So dendrite, soma, axon. So that's what you should keep in your head for this. And going back a little bit, just in terms of connection, because I don't want you to feel like this is the only route of communication, which is axon to dendrite. There are also connections that are axon to axon, dendrite to dendrite, axon to cell body. But again, we're going to focus on what we typically think of and what are most common connections, which are axon to dendrite. So we discussed communication within. Okay, so the action potential within. But now we're going to talk about how information is sent from neuron to neuron, which creates that beautiful highway system in the picture that we just saw. So in most cases, action potentials are not transmitted from one neuron to another neuron. They're within an individual neuron. And information from neuron to neuron is transmitted at synapses. Okay, so I had said that communication between neurons is also known as synaptic transmission. Typically, a chemical neuron, neurotransmitter, carries information from a presynaptic cell to a postsynaptic cell, which we're going to talk about now. So just some definitions to get us all on the same page. A synapse is essentially a gap. So if you look at the picture on the right, which kind of looks like two monsters kind of like going at each other, the gap in between is the synapse. There are two types of connections and two types of synapses. There's chemical, it's what we typically think of. It's when a neurotransmitter is being sent from one cell to another. And then electrical, which is not as common, but really important for survival. And we're going to get into that a little bit later. So presynaptic neuron is the neuron that's transmitting the signal. So if you look at the picture, it's kind of that monster on top. That's the presynaptic. And then the cell that's catching it, that neighboring cell, is the postsynaptic, but they can be used interchangeably. So most neurons have both presynaptic, they can act as presynaptic and postsynaptic. So the presynaptic cell was the postsynaptic cell at one point. So if we look at the diagram on the right, and you look at the one on top, that was a presynaptic or postsynaptic cell at one point when it received information from a neighboring cell, but now it's acting as, it was a postsynaptic cell, now it's acting as a presynaptic cell to send information to a postsynaptic cell. Then that postsynaptic cell will act as a presynaptic cell to send information to a postsynaptic cell. I just said that a lot of different times, but essentially the term can, each cell can act as both. But when you're looking at communication from one individual cell to another, we're looking at the information that's being sent is from the pre, and the information that's being received is from the post. So essentially, that top monster is sending neurotransmitters to that bottom monster. The neurotransmitters are the body's chemical messengers, and they give information about what is supposed to happen to that bottom cell. So here, what I tend to do is go into like kind of a simplistic picture and then go into the individual components and get a little bit more complex as I go along. But just to start us off, let's talk about the individual steps of communication between neurons. So the presynaptic neuron will synthesize and package the neurotransmitter in synaptic vesicles, which are located in the axon terminal. So those little button areas that we saw, if we go back to the beginning, in the axon. The arrival of an action potential in a cell causes the release of a neurotransmitter. Just to clarify, dopamine, serotonin, norepinephrine, those are all examples of neurotransmitters. The neurotransmitter is going to diffuse or go across that synaptic cleft or synapse or gap, like to use a lot of different terms for the same thing. And they're received by the postsynaptic cell, or like the bottom monster. And then just as quickly as they come, they leave. So you can't have a chemical continuously providing action on that postsynaptic cell, because things can go awry. And so then there are different systems put in place in which the neurotransmitter is removed from that space. So there's different, like I said, there's different ways in which it can be removed. One example is reuptake. So many drugs for mental health conditions work in this way. If you've ever heard of selective serotonin reuptake inhibitors, the name is exactly what it is, or what it's doing. So it's keeping the serotonin in the postsynaptic cell, taking action in the postsynaptic cell longer. So it's prohibiting like a fast reuptake so that there's more action of serotonin taking place in that cell. So it's a reuptake inhibitor, not that it's inhibiting indefinitely, but it's inhibiting for a longer period of time. All right, so I mentioned that there's different types of synapses. There's chemical synapses and there's electrical synapses. So in the green, and I know this is kind of small, but in the green, that's an example of a chemical synapse, and there's a space there, and information is sent through that space. And the blue is an example of an electrical synapse. So a chemical synapse involves the release of chemical messengers known as neurotransmitters. They carry information from the presynaptic or the sending cell to the postsynaptic or the receiving cell. Electrical synapses are less common, but they're essential. They're a faster action because they don't have that gap. So there's a direct connection between the presynaptic and the postsynaptic cell. The connection takes the form of what's called a gap junction, which allows ions to flow directly from one cell or another. So we're leaving out that middle space. So we started with communication that takes place within an individual neuron. And it's really important for students to understand that so that they can understand this step, because it all goes together. This is where we start. So I mentioned that there are chemical and there are electrical synapses. Oops, I was distracted by the chat again. I need to stop doing that. Okay, I get excited about the little blinky. Okay, so action potentials start this process here. And so information is received from presynaptic to postsynaptic. We're going to talk about how an action potential would take place or not. But let's start with, say, an action potential does take place. So there's enough stimulation for an action potential to take place, and it reaches the axon terminal. So that end part of the axon, it depolarizes the membrane. So depolarizing, again, remember, making more positive, and opens voltage gated sodium channels and allowing the sodium channels to enter the cell, further depolarizing that presynaptic membrane. This allows for calcium channels to open. Calcium channels aid to help with the release of neurotransmitters. They enter the cell and initiate a signaling cascade that causes synaptic vesicles containing the neurotransmitter to fuse with the synaptic membrane, the presynaptic membrane. Okay, so we're still within the presynaptic cell. Fusion of the vesicle with the presynaptic membrane causes neurotransmitter to be released into the synaptic cleft, which is that space in between the presynaptic with the postsynaptic neuron. Neurotransmitter will diffuse across the cleft and bind to the receptor proteins on the postsynaptic membrane. So I said that at the end, we're going to talk about receptors. Again, everything is going to come together here, but essentially it's going to be what receives that neurotransmitter from the presynaptic to the postsynaptic cell. that binding of the neurotransmitter, whatever it is, it could be serotonin, causes ion channels on the postsynaptic membrane to open. Okay. And then what happens from here? So there's an action that takes place from here. And then before that action takes place, the neurotransmitter is taken out of that postsynaptic cell, even though it's already created action. And we're going to talk about how that takes place as well. But I want to talk about the action that takes place on the postsynaptic cell once it gets, the neurotransmitter gets there.
Hey, Laurie, I apologize for interrupting. I just want to give a quick 10-minute warning just so we have enough time at the end for questions.
I'm glad you did that because I will talk for 3 hours.
I was just going to say, probably around the 5-minute mark, you'll see me cut in again and we can start questions then.
Okay, please do cut in again because again,
Awesome.
I will talk for 3 hours. All right, thanks, Brian. Okay. When we talk about action potentials, I said it depends if there's enough stimulus to reach a threshold of excitation. And so the threshold of excitation is reached, then the action potential will fire. So we're going to go back in time here. So we have two different mechanisms that can take place on the postsynaptic cell. We have excitatory postsynaptic potential, or EPSP, which is the depolarization of the presynaptic membrane, which makes the postsynaptic membrane more likely to fire an action potential. And then we have IPSP, or inhibitory postsynaptic potential, which is a hyperpolarization of the presynaptic membrane, which makes the postsynaptic neuron less likely to fire an action potential. So excitatory, more likely; inhibitory, less likely. So if we look at an example of an EPSP, we can think of the neurotransmitter acetylcholine, which is really important for muscle movement. It's also important for learning and memory. It's released at the synapse between a nerve and muscle called the neuromuscular junction. It causes postsynaptic sodium channels to open and for sodium to enter the postsynaptic cell, which causes the postsynaptic membrane to depolarize. So that is called an EPSP. That postsynaptic cell is depolarizing, more likely to fire an action potential. On the other hand, if you've ever heard of GABA, which is what's essentially increased in the brain if you take like a benzodiazepine, like a Xanax, it helps with anxiety. It has inhibitory effects. A release of a neurotransmitter at inhibitory synapses like GABA causes inhibitory postsynaptic potentials, or a hyperpolarization of the presynaptic membrane. All right, let's try to pick up the pace a little bit here. So how do excitatory postsynaptic potential and inhibitory postsynaptic potentials interact? Basically, a postsynaptic cell is going to add together, integrate all of the signals that it's getting. So all the excitatory and all the inhibitory signals, and decide whether it's going to fire an action potential. This can take place by way of spatial or temporal summation. Spatial summation is the integration of postsynaptic potentials that occur at different locations, but at the same time. So if you have multiple neurons together releasing neurotransmitters like acetylcholine, an excitatory neurotransmitter, and that can be enough to exceed the threshold on the postsynaptic neuron. So just an example there, let's suppose that there's excitatory synapses are on different dendrites of the same postsynaptic neuron, but neither synapse can produce quite enough EPSP to bring the membrane potential to threshold of excitation where the action potential is triggered. However, if you have both EPSPs that take place at the same time, it could sum or add up enough to bring the membrane potential to the threshold. On the other hand, if you have an IPSP with two EPSPs, it may prevent the membrane potential from reaching there because you have inhibitory effects too. Temporal summation occurs when one presynaptic neuron releases neurotransmitter many times over a period of time to help the cell reach threshold of excitation. So that's the same cell over and over, and spatial is an integration of many different signals. So I mentioned that once there's action that takes place on the postsynaptic neuron, there's a removal of the neurotransmitter from the cleft. There's 3 ways in which this can happen. So there's diffusion, in which the neurotransmitter will diffuse from the synapse into fluid tissue from the synapse and get absorbed by what's called glial cells, which are the other type of brain cell in addition to neurons. There's degradation. So there can be a helper enzyme, essentially, that can help break down this neurotransmitter. Example, there's something called acetylcholine X-Rays. That is an enzyme that breaks down acetylcholine, which I've mentioned. This can help make it inactive, so it's not constantly providing action on that postsynaptic cell. And then it can be recycled or reuptaken. So this happens to classical small molecule neurotransmitters, which we're going to talk about. So like glutamate, dopamine, serotonin, epinephrine, norepinephrine. And so essentially there's helpers to help that neurotransmitter go back to the presynaptic neuron. Quickly, we're not giving them that much attention, but electrical neurotransmitters, it's when the presynaptic and postsynaptic membranes are physically connected. 5 minutes.
Yeah, I do apologize again, but just it is 5 minutes. If you want to jump to the Q&A now, we can. So it's yeah. But
I actually think we can. I think we're almost done. I can go quickly.
Sure.
The
That
next
sounds
5
good.
minutes.
Yeah, that's fine.
You're like, stop talking. Okay.
No, you're good. So as long as you address the question.
You're right. So electrical synapses are much more reliable. They're fast and they can be really useful if you're in a situation like you see a bear in the woods and you need to escape from that. You need faster action of the brain, and so you're able to take action to, say, take fight or flight. They're not very flexible though. They don't allow for accuracy and precision, so they're not as numerous or commonly used as chemical synapses. So I want to go past, so just quickly, there's multiple different types of receptors. There's more fast acting receptors, which are called ionotropic receptors. And then there's receptors that need a little bit of help, and those are called metabotropic receptors. So they're not themselves ion channels, but they need help in order to get information from one place to another. Ionotropic receptors are the fast ones. They undergo a change in shape when a neurotransmitter binds. So this is on the postsynaptic cell. There's either an excitatory or an inhibitory effect. And it typically produces a quick physiological change, and the neurotransmitter is removed quickly after binding to that receptor. Metabotropic are slower acting receptors. A protein to which the neurotransmitter binds is not the ion channel. It depends on the activation of several molecules inside the cell, and often involves a second messenger pathway, so a helper cell. It's much slower at signaling. So quickly, I had mentioned neurotransmitters many times, so I just want to brush on those a bit. It's essentially the body's chemical messengers. So they're molecules used by the nervous system to transmit messages from between neurons and from neurons to muscles and glands. A single neurotransmitter may have dozens of different receptors and take place in various areas of the brain. And there are many, many different types of neurotransmitters. Many are still being discovered. So conventional neurotransmitters all share basic features. They're stored in the synaptic vesicles of the presynaptic cell. They get released when calcium enters the axon terminal, and they act by binding to receptors on the postsynaptic cell. Not going to go into all of these, but very common conventional neurotransmitters, checking the time, are small molecule neurotransmitters. I mentioned acetylcholine, there's amino acids, and biogenic amines. So acetylcholine, because I talked about it a lot already, it's a very common neurotransmitter, essentially for vitally important for muscle stimulation, memory, formation, and learning. There's amino acids, glutamate, which is the most common brain neurotransmitter and has an excitatory effect. It's important for learning and memory. And then there's GABA, which is the main inhibitory neurotransmitter in the brain. This goes back to EPSPs and IPSPs. So some can excite a cell to take action, and then GABA inhibits the cell to take action. What we usually think of when we think of neurotransmitters are in this category. They're active in both the central nervous system, so the brain and spinal cord, and throughout the rest of the body. They play a central role in nervous system disorders and treatments. Some examples of these are norepinephrine, epinephrine, dopamine, which is that feeling when you just want to keep eating cookies because it gives you so much pleasure. You eat one and then another. And serotonin, which is a neurotransmitter that contributes to feelings of well-being and happiness. I did it. It's 3:45. I didn't talk to you for 3 hours. So thank you for listening to me talk about neuroscience. I am happy to answer questions, and I have included my email.
That's great. Thank you very much for that. And we'll go into a couple of quick questions, and then we'll have to turn it over to the next presentations. But so the first question that we saw, and I think you hit on it a little bit, is how do you feel about your researchers who believe and work with fMRI to read thoughts?
I guess other people could disagree with me, but there's not a way to read thoughts. I mean, fMRI is looking at essentially the oxygen. So where your brain is active at a given moment, either at rest or if you're participating in some sort of activity. So it's really telling us what part of the brain is responsible for whatever is taking place. So it gives us some insight into what's happening in the brain, but I think it would be a very far stretch to say that it is helping us to read minds, just like I gave the example of clinical psychologists having the ability to read minds because they understand human thinking based on literature.
Awesome, thank you. And I know since we're running out of time, I'll ask one more quick question right here. How many approximately neurotransmitters have been identified in humans?
Oh, I'm not sure about that. That's a really good question. Thank you for asking me a really, really tough one. But I want to go. I can ask another one.
Huh? There's a couple more in here. I can ask another one too.
No. That's a really good question. And I'm not exactly sure. There's I want to say that we're at thousands. My screen went down.
And I will pick out one more for right now. Have you worked with neural networks directly or indirectly with mathematicians or computer scientists?
Yes. So, well, I guess it depends how you look at it, but I used to work at Washington University in St. Louis, and we did brain mapping there. So I was actually trained to help work on the brain mapping, so able to look at those highway systems within the brain. And we also worked with computer scientists to help map the brain and essentially to generate that picture that I showed you before of all of the different colors.
Perfect, that sounds good. And I know we are past the time limit, but if you'd like to answer a couple more questions, we can. It's up to you and the attendees. So,
well, how about this? I am going to give you, because I overtalked, I am more than happy to chat with anybody. I love to talk about this stuff. This is my email. You definitely send me an email if you have any questions or you want to talk about science. I'm happy to, and we can go from there.
Perfect. That sounds great. And for all the attendees that are still here, first and foremost, I'm going to launch a quick 3-question poll on the presentation that we just watched. Please take a minute to give us some feedback on the presentation and your experiences today. And it'll probably take you no more than a minute to complete the poll. I'm going to release the poll right now.
All right, thank you so much for attending, everyone, especially for spending a Friday afternoon with me listening to science, and I hope everyone has a really nice weekend.
Thank you very much. We really appreciate it. And just on behalf of everyone here at Hawks Learning, we thank you, Laurie. We thank all of our presenters and attendees for being a part of IES. The recordings of this year's sessions will be provided to everyone in the next couple of days. And if you haven't already, don't forget to swing by the exhibit hall to say hi to someone on our Hawks team. If you view a quick 5-minute demo, and then you will be entered into win an hourly giveaway of a choice of a smart pen, standing desk, or a $100 Amazon gift card. Thank you again for joining us at IES. We really appreciate it.